Hi, my name's Michelle. We're going to go over the introduction to the human body. Let's get started. All right, and these are the things we're going to go over. All right, so first off, what is anatomy? Anatomy is the study of the body's regions. There's two types. There's gross or macro, which is a study of the larger body structures, like your organs, such as your brain. Then you've got micro, which is going to be on a microscopic level. Then you've got regional anatomy, which is going to be studying, for an example, the abdomen, how all the muscles, the nerves, and the blood vessels and the structures work together to serve that particular region. Whereas systemic anatomy is going to be a more discrete body system. So it's going to be looking at, for example, the muscular system is going to include all of the skeletal muscles of the body. And then we are moving on to physiology, which is a scientific study of the chemistry and physics of the structures of the body. Okay, now we have the four functions of human life. Without them, we can't survive. And that's going to be oxygen, nutrients, a set temperature range, and a set atmospheric pressure. Side. Then we've got our levels of organization. So our atom to atom, we have when you have three or more atoms that bond together, that's going to form a molecule. Then your molecules are going to join together. That's going to form your fluid and your organelles of your cells. Then your cells are going to combine and form tissues. Then your tissues are going to combine to make organs. When you have two or more organs working together, that forms an organ system. And then once you have multiple organ systems working together, you have your organism or your atom. Homeostasis is going to be the body's process of maintaining a state of balance to keep us healthy, helps prevent us from getting sick, and it does that through maintaining a set body temperature, blood pH, blood glucose, and blood pressure. So the way our body is going to maintain homeostasis is through positive and negative feedback loops. So negative is going to sense when something is out of balance, and it is going to send a corrective action. So if we overheat, our body is going to start sweating to correct that overheating process. Whereas with positive feedback, our body is going to intensify until it gets the desired outcome. So an example would be childbirth. It's going to continue to intensify contractions until labor, until childbirth. Next, we have the body systems, which is your integumentary system, your skeletal system, your muscular, nervous, endocrine, cardiovascular, lymphatic, respiratory, digestive, urinary, and reproductive systems. Then we have some regional and body terminology. We also, not included on this slides here, we have our anterior, which is going to be the front. It's also called ventral. You can think like a vent, the mouth. Then you've got your dorsal or your posterior, which is the backside. Superior is going to be the top of the head. Inferior is going to be down at the feet. You've got your distal and proximal. Distal is going to be further away. Proximal is going to be closer. And of course, you've got your different anatomical language here that you can see. We've got our right hypochondriac area, which is going to be like below the breast and around the rib cage area. Your epigastric is going to be the center upper part under the breasts. And then your left hypochondriac is, of course, the same as the right on the left side. Right lumbar region is going to be kind of just like where the end of the rib cage is to just above the hip, where your right iliac inguinal region is going to start. The umbilical region is going to be the center. Your left lumbar, of course, and your left iliac, that's going to be the same as the right side. The iliacs are on the right and the left. That's going to be your hip area. And then your hypogastric is your pubic area. It's below the belly button. Then you've got your right, left upper quadrants and your right and left lower quadrants. And here we've got our different types of medical imaging. We've got x-rays, which is a two-dimensional image used to look mainly at the bones. CT scan, which is non-invasive. It's cross sections. Your MRI, which is used to look at soft tissues and body images. It uses magnetic fields. Your ultrasound is going to use high frequency sound waves, and then we've got our PET scan, which uses radiopharmaceuticals to emit short-lived radiation to detect abnormalities like cancer. And then this, of course, is going to be a comparison of the four main types. You can see here that the CAT scan has the most radiation. MRI and ultrasound have no radiation. X-ray has minimal radiation, and it kind of goes over what they're used for commonly and what images are going to be the best. MRI is going to probably be the best imaging. On to chapter 2, the chemical level of organization. Next, we've got the building blocks of life. So you've got your atom, which is the smallest unit of matter. All elements are made up of atoms. Atoms are made up of smaller subatomic particles. So you've got your proton, which is positively charged. Neutrons are neutral. Electrons are negatively charged. Molecules are two or more atoms that are joined together. CHON is carbon, hydrogen, oxygen, and nitrogen. Those are the most abundant elements in the human body. And then valence electrons are the atom's outermost electron shell. Life. We've got our periodic table of elements. It's divided into columns. Like categories are together, so all of your gases are together, your heavy metals are together, and they are in various columns based off of their atomic number and mass. Their atomic number is the number of protons in the nucleus of an atom. Your mass number is going to be the sum of the numbers of your protons and your neutrons in the nucleus. Then we have ions. Ions are atoms that have a positive or a negative charge. A cation, which has a plus in it, is a positive charge. An anion is a negative charge. Then we have covalent bonds, which is where basically the molecules share electrons and it's mutually stabilizing. It's a mutual relationship. You've got nonpolar and polar. Nonpolar is when the electrons share things equally, and then the polar, they have different electronegativities and they share. Hydrogen bonds are when there's an attraction between two atoms, but they already participate in another chemical bond. So you can see that the hydrogen bond is broken, whereas the covalent bond is more solid. We have ATP, which is adenosine triphosphate. That's used to power the cellular process. So our body energy is released by the breakdown of the ATP through hydrolysis, and it's used to power cellular. Okay, then we have our chemical reactions, and this is when a chemical reacts with another substance and it acts as a solvent. Metabolism is a chemical reaction. It's where the body cells change food into energy. And then we have other types, which is synthesis reaction, decomposition, which is breaking down, synthesis is joining together, exchange is rearranging things, and then catalyst speeds things up. Then we have organic compounds. These contain both hydrogen and carbon. These are important because these are abundant throughout the body and it's what we live on. We need it to survive. Your proteins are organic molecules made up of amino acids and they're linked by a peptide bond. They're both functional and structural. Nucleic acids is going to be like your DNA. It's the building block of who you are. They're naturally occurring chemical compounds found in all cells, and it's vital to all living organisms. You can't be you without DNA and RNA, which is a replication. Carbohydrates are fancy sugars, and then we've got lipids, which are your fancy fats. For the carbohydrates, there's monosaccharides, disaccharides, and polysaccharides. For your lipids, you've got glycerides, sterols, and phospholipids, and prostaglandins. Organic compounds and inorganic compounds, they're different. One has the hydrogen and carbon, the other one does not, but they both are chemical compounds made up of chemical or a combination of elements. You have your um, inorganic compounds are going to be things like water. Water is extremely important to the body. Colloids is a mixture of a somewhat heavy solution. Think of like milk and cream. Your thyroid hormone is stored in a colloid. A suspension is kind of like a snow globe. So like we use it for a SED-rate test to measure how quickly your blood cells um, separate from the plasma, so the watery substance of your blood. So like if you shake up a snow globe, those little particles are going to float and then they're going to sink to the bottom. Peptide bonds are covalent bonds between two amino acids that form by dehydration synthesis, which is a chemical reaction where a molecule is reacting while water molecules are being taken out. Hydrolysis is a water molecule disrupts a compound, so the water gets split into a hydroxyl. So it's one part bonds with the hydrogen atom and the other part with the hydroxyl group. Then we have our salts, our acids, and our bases. So a salt is going to be formed when ions form an ionic bond. So one atom is going to give up one of its electrons, one or more of its electrons, and it's going to become positively charged. Acids, a substance that it releases hydrogen ions, so it's hydrogen positive in a solution, and then your base is going to be the substance that releases hydroxyl ions in the solution. A buffer is a solution of weak acid and its conjugate base. Bicarbonate, that's the main buffer in our blood. Our blood pH ranges between 7.35 to 7.45. And it helps reduce the acidity from carbon dioxide consistently being released into our bloodstream. Okay, now we are on to our last chapter, which is the cellular level of organization. These are the things we're going to be going over. We have our human cell, which is made up, the outer part is the cell membrane. It's a double layered phospholipid membrane. It separates the outside and the inside of the cell. It's also called the plasma membrane. It has a hydrophilic head, which means that it likes water, and then a hydrophobic tail, which is afraid of water. And it regulates and transports materials entering and exiting the cells and provides structure to the cell. Then we have our cytosol, which is the jelly-like substance contained in the organelles. Your endoplasmic reticulum, there's two types. You've got your rough, which is dotted and embedded with granules. This is organelles called ribosomes, and these give it the bumpy appearance. Ribosomes are an organelle that serves as a site of protein synthesis. Then your smooth ER does not have the ribosomes, and so therefore it is smooth. This is just another picture. We also have the Golgi apparatus, which is kind of like the post office, or like, I guess, Amazon Prime these days. It sorts, modifies, and ships off products from the rough ER. It produces lysosomes, which is an organelle. It contains an enzyme that breaks down and digests unneeded cellular components, such as damaged organelles. It performs autophagy, so it's self-eating. You can see in the little comic over here. And then the lysosomes also perform a programmed cell death by explosion. You've got the mitochondria, which is the energy transformer, so the Optimus Prime, ATP, which is what stores your energy. Your peroxisomes is a membrane found in the organelles, and they perform lipid metabolism, so they neutralize free radicals. They're like scavengers. Then we've got a cytoskeleton. This is the protein that makes up the structure of the cell. Then we've got the nucleus, which is the control center. Then we've got multinucleated cells. Here we have the nucleus and the different parts. We got your nucleus envelope, the nuclear lamina, the nucleolus, nucleoplasm, chromatin, endoplasmic reticulum, the ribosomes that give the rough ER its rough edges, and your nuclear pores. Next, we have our cell cycle. So mitosis is the division of genetic material in which the cell nucleus breaks down into two new nuclei. Um, the stages of the cell division is the prophase, then you've got the metaphase, the anaphase, the telophase, and then finally cytokinesis, which is where the cell actually splits into two. And here we just have some important vocabulary, um, some notable notes that are worth looking over. Cellular transport, there's passive transport, which is how things move in and out of the cell without using energy. They spread things out. So an example of this would be reabsorption of nutrients by the intestines. They separate them through the solid waste, and then they transport the nutrients through the intestinal membrane. There's active transport where the cells concentrate closer together and they move together. So an example of this is going to be an uptake of glucose in the intestines of the human body. Osmosis is the diffusion of water. Water moves because the molecules don't fit. This is endocytosis is how things get into the cell. Then you've got your exocytosis. This is how things move out of the cell. I think endo like enter and exo as an exit. Then you have your examples of cellular hydration. So if you're not getting enough water or fluids, your cells are going to become dehydrated. Water's moving out of the cells. It's going to need to go elsewhere throughout the body to be used. If it's isotonic, then it's balanced hydration. Hypotonic is too much hydration. So you can see the cells look overhydrated. They look like they're swollen. DNA stands for deoxyribonucleic acid. It's a molecule that carries genetic information from the development and function of an organism. Its components are adenine, thymine, guanine, and cytosine. They're complementary, so the A's and the T's go together, the adenine and the thymine, and the guanine and the cytosine go together. So replication is going to be how DNA is made, and it's where a leading strand will move towards the zipper and the lagging strand moves away from the zipper. Initiation is going to be when the two strands separate and the special enzymes like the helicase untwist and separate the two strands. Then you're going to have your elongation, so the two strands will become a template along with the new complementary strand. Then you've got your protein synthesis. Synthesis again is where things are joining up. So you've got your carboxyl group and it forms a peptide bond with a polypeptide chain. Transcription is going to be turning DNA into RNA, so it's the same but different. Makes me think of multiplicity, so it's like a copy of a copy. And then you've got your transcription and translation. So they both have cytosine, guanine, uracil, and adenine nucleotides, but translation is going to be, it occurs in the ribosomes, and it's an RNA to a peptide, or excuse me, it's RNA to a protein. Transcription is turning DNA into RNA. And then termination is when you get your two original strands of your DNA and they're bound and they're finished, then the body stops the process. And lastly, we have cell differentiation. So it's where the unspecialized stem cells turn off their proteins and they create more specialized stem cells, and they become further specialized. So you've got your embryonic stem cell, and then it becomes an ectoderm, a mesoderm, or an endoderm. So ecto is outer, meso is middle, endo is inside. And then so you've got your skin and nerves are going to be your ectoderm. Your mesoderm is going to be things like your bone and muscles, and then your endoderm is going to be like your liver and your thyroid. And that completes our presentation. The last 2 slides are just my references. Thank you so much for your time. Have a great day.